Welcome back. Myself, Dr. G. Prasanna, working as an assistant professor in the Department of Genetics and Plant Breeding at School of Agriculture, SR University, Warangal. Today, we'll discuss about the most important topic, that is breeding for wheat, oats, and barley crop. So, here are the lecture outlines. Here we'll discuss about the centers of origin of all the crops, that is wheat, oats, and barley. And next we'll discuss about the distribution of the species, breeding objectives, and next breeding methods, which includes both the conventional breeding methods as well as the innovative breeding methods. First, we'll discuss about the wheat crop. As we know, the scientific name of the wheat is Triticum aestivum. It is having the chromosome number 2n equal to 6x equal to 42. Here, the 6x is nothing but it is a hexaploid species. So, the chromosome number is 2n equal to 6x equal to 42. As we know, the wheat is the most important staple food crop in the world. It gives around one third of the total production, followed closely by the rice. Next, it is a it is a staple food of one by third of the world population. Every month of the year, a crop of the wheat is harvested somewhere in the world. Next is place of origin. The place of origin of wheat is, it is Southwest Asia. The centers of origin for different wheat species are given below. So here we are having the three different types of species in wheat. That is a diploid species, tetraploid species, and hexaploid species. For diploid species, it is having the chromosome number 2n equal to 14, and for tetraploids, it is having the chromosome number 2n equal to 28, and for hexaploid, it is having the chromosome number 42. So, the scientific name of the wheat is, it is Triticum aestivum. It is having the chromosome number 2n equal to 6x equal to 42. Here the 6x, it is a allohexaploid. So it is represented as a 6x, and it is having the chromosome number 42. And this Triticum aestivum is also called as a common bread wheat. Um, Next, as we know, the wheat is the most important staple food crop in the world. It gives around one third of the total production, which was followed by the rice. And next is, it is a staple food crop of one by third of the world population. Every month of a year, every month of a year, the crop of wheat is harvested somewhere in the world. So it is considered as one of the most important staple food crop in the world. And next, coming to the place of origin, the place of origin of the wheat is, it is Southwest Asia. And here we are having the three different types of wheat species based on the ploidy. Here we are having the diploid species, tetraploid species, and hexaploid species. For diploid species, it is having the chromosome number 2n equal to 14. For tetraploid species, it is having the chromosome number 2n equal to 28. And for hexaploid, it is having the chromosome number 2n equal to 42. Next, coming to the cultivated species. In wheat, we are having the two cultivated species. The first one is a common wheat, that is Triticum aestivum, that is a common bread wheat, Triticum aestivum, and durum wheat, that is a Triticum turgidum. These are the two cultivated species in the wheat. That is a common wheat, that is a Triticum aestivum, that is hexaploid, and Triticum durum wheat. This is a Triticum turgidum. It is a tetraploid species. So these are the two cultivated species in the wheat. Next, common wheat is, generally the common wheat is a hexaploid, because it is having the chromosome number 2n equal to 6x equal to 42, and whereas the durum wheat is tetraploid, it is having the chromosome number 2n equal to 4x equal to 28. So these are the we are having two cultivated species, that is common wheat, that is Triticum aestivum, and durum wheat, Triticum turgidum. And this common wheat, that is Triticum aestivum, having the chromosome number 2n equal to 6x equal to 42, whereas the durum wheat, it is having the, which is a tetraploid, it is having the chromosome number 2n equal to 4x equal to 28. Next, bread wheat is more adapted than compared to the durum wheat, and generally the common bread wheat is used for making the bread. cakes, noodles, cookies, and chapatis, whereas the durum wheat is used mainly for making the marconi and some flat breads. And in wheat, there are 16 wild species, out of which two are diploids. There are two, they are having the chromosome number 2n equal to 14, and seven are tetraploids. They are having the chromosome number 2n equal to 4x equal to 28, and three are hexaploids, which are having the chromosome number 2n equal to 6x equal to 42. So this is about the cultivated species. This we are having the two cultivated species in the wheat, that is a Triticum aestivum and Triticum durum. Triticum aestivum is the hexaploid and Triticum durum is the tetraploid. Here hexaploid it is having the chromosome number 42, whereas tetraploid having the chromosome number 28. Here out of two cultivated species, the bread wheat is more adapted than the durum wheat, and the bread wheat is mostly used for the making the breads, cakes, noodles, cookies, and chapatis, whereas durum wheat is having the less usage, which is used for making marconi and some of the flat breads. And here the wheat is having around 16 wild species, out of which two are diploids and seven are tetraploids and three are hexaploids. The diploids are having the chromosome number 2n equal to 14, and seven are tetraploids, which are having the chromosome number 28, and hexaploids are having the chromosome number 42. Next, coming to the utility based on the quality, they are divided into two types. The wheat is divided into two types, that is hard wheat and soft wheat. Here, the hard wheat is the Triticum durum, and soft wheat is the Triticum aestivum or Triticum vulgare. So, yeah, as we discussed, the Triticum durum is it is a tetraploid having the chromosome number 28, and it is a durum wheat, and it is also called as durum wheat. And the in case of Triticum durum, the kernels are very small, and they are having the protein content around 13 to 16%. And this hard wheat are low yielders, and they are more glutinous. They are having the more gluten content. And this hard wheat is used for making the marconi and as well as the chapatis. And next, coming to the soft wheat, that is Triticum aestivum and Triticum vulgare. Here, the soft wheat it is a hexaploid. It is having the chromosome number 2n equal to 42, and it is also called as a bread wheat. And here the kernels are very large, and these are having the this soft wheat is having the protein content around 6 to 11%, and these are high yielders when compared to the hard wheat. And these are having the these are non-glutinous. They are having no gluten. And next, these are used for making the cakes, pastries, and cooking. So this is a based on the quality. They are divided into two types, that is hard wheat and soft wheat. Hard wheat is the Triticum durum, and soft wheat is the Triticum aestivum or Triticum vulgare. So here the Triticum durum is a tetraploid, and here the soft wheat is a hexaploid. And the tetraploids are having the chromosome number 28, and hexaploids are having the chromosome number 42. And here the durum wheat, and this is known as the durum wheat, and this is known as the bread wheat. And here for hard wheat the kernels are small, and for soft wheat the kernels are large. And for hard wheat the protein content is 13 to 16%, and for soft wheat it is 6 to 11%. And generally this hard wheat is low yielders, whereas the soft wheat are high yielders. And they are this hard wheat having are they are a gluten, they are having the gluten content. The protein in wheat is nothing but gluten, that is, so the hard wheat are glutinous and the soft wheat are non-glutinous. So here the hard wheat is used for making the marconis and the chapatis, whereas the soft wheat is used for making cakes, pastries, and cookies. And next, coming to the origin of species, here in wheat we are having the origin of the diploid wheat. Here we are having the tetraploid wheat and hexaploid wheat. We'll discuss how these three species were evolved. So first we'll discuss about the origin of the diploid wheat. First is that is the Triticum boeticum. And this Triticum vomiticum was obtained through natural mutation and selection, and through that the Triticum monococcum was obtained. That is a cultivated diploid. It is having the chromosome number 2n equal to 14 and genome of capital A, capital A. This Triticum vomiticum is the ancestor for the all the cultivated wheats. And next, coming to the This is the origin of the diploid species, that is the Triticum boeticum through natural selection and mutation. The Triticum monococcum was obtained. It is a cultivated diploid. It is having the chromosome number 2n equal to 14, and the genome is it is represented as a AA. And this Triticum boeticum is the ancestor for all cultivated wheats. So this is the origin of diploid wheat. Next we'll discuss about the origin of tetraploid wheats. First, here the Triticum monococcum, as we discussed, this is a cultivated diploid. It is having the chromosome number 2n equal to 14 and genome of AA. So this was crossed with unknown species, that is Agelopus speltoides. It is also diploid. It is having the chromosome number 2n equal to 14 and it is having the genome of capital B, capital B. When we'll cross this Triticum monococcum and unknown species, we got the F1 hybrid, which is a diploid and sterile. And so here the genome is capital A and capital B, and it is a diploid. Automatically the chromosome number is 2n equal to 14. So again, this F1 hybrid is undergone for chromosome doubling with colchicine treatment. It will result in the tetraploid, that is a Triticum turcidum. It is an amphidiploid or allotetraploid, and it is having the chromosome number 2n equal to 28, and the genome is capital A, capital A, capital B, capital B, and it is a fertile. So this is the origin of tetraploid species. The Triticum aestivum is crossed with unknown species, that is Agelopus speltoides. We got the F1 hybrid, which is a diploid. So it is a sterile. So the chromosome doubling was done with the colchicine treatment to so that we got the Triticum turcidum, which is an amphidiploid or allotetraploid. It is having the chromosome number 2n equal to 28, and the genome is capital A, capital A, capital B, capital B, which is a fertile. So these are the two origins of the diploid species, and next is tetraploid species, and next we'll discuss about the hexaploid species. This is the most important origin in case of wheat. So Triticum turgidum. So this Triticum turgidum, this is a tetraploid species, which is which is having the chromosome number 28, and the genome is capital A, capital A, capital B, capital B. It was crossed with Triticum tauchi, that is a diploid having the chromosome number 2n equal to 14, and the genome is capital D, capital D. When we cross this tetraploid with hexaploid, we'll got the F1 hybrid, that is triploid, that is 2n equal to 3x equal to 21, that is A, B, and D genome we got. So it had this F1 hybrid, that is tetraploid, is sterile. So the chromosome doubling was done with the colchicine treatment to get the fertile hybrid, that is a hexaploid, that is a we got Triticum aestivum, that is a common bread wheat, that is a hexaploid, that is having the chromosome number 2n equal to 6x equal to 42. So here the genome gets doubled, that is AA, BB, and DD. This is a fertile. So these are the three. This is the origin of this wheat species. How the diploid wheat was obtained, and how the tetraploid species was obtained. And next is how the hexaploid species was obtained. And next, coming to the breeding objectives. Breeding objectives here, in each and every crop, the yield is the most important breeding objective. So the yield in the wheat depends upon the number of heads per unit area, and the number of the grains per head, and the average weight of the grain. So depending upon these three parameters, the yield will be dependent, that is number of heads per unit area, and number of grains per head, and next is average weight of the grain. These three parameters will decide the yield in case of wheat. And next is breeding for non-lodging varieties. Generally, for non-lodging type, we are also having the semi-dwarf wheat varieties in case of wheat. Semi-dwarf wheat we are having, and they are having the dwarfing genes, that is Norin 10 in case of wheat crop. So we have to breed the varieties which are having non-lodging varieties. And next is breeding for disease resistance. Generally, we are having the major disease in case of wheat is it is rust. We can see the stem rust, leaf rust. So So we have to breed the varieties which are resistant to all three different types of rust. And next is breeding for the insect resistance. And we are also we have to develop the varieties which are resistant to the different pests which are present in the wheat. And next is breeding for the quality. Generally, quality is nothing but here we have to breed the varieties which are with which based on our requirement we have to breed and develop the varieties like soft wheat and bread wheat. This is about the breeding objectives. We are having the five important breeding objectives in case of wheat, that is high yield, and next is breeding for non-lodging varieties, and breeding for disease resistance, and breeding for insect resistance, and next is breeding for the quality. Coming to the breeding methods, so the first breeding method is introduction. Here we the semi-dwarf wheat varieties which were introduced from the Mexico, and next Sonara 63, Sonara 64, and Mayo 64, and Lerma Roja. These are the semi-dwarf wheat varieties which were introduced from Mexico. And next is pure line selection. Earlier, P4, P6, and P12, they are evolved at PUSA Institute, which are the result of the pure line selection from the local population. Next is hybridization and selection. We are having different types of hybrids in case of wheat. Next is mutation breeding. The mutation breeding is Dr. Swaminathan has conducted a mutation breeding. He has done more work on this mutation breeding. He worked with gamma rays, and as a result, he developed the Sharbati Sonara with increased protein content. And next is development of the multi lines. This multi lines are nothing but these are a mixture of several pure lines which are having the similar plant height, like similar flowering, that are nothing but multi lines. So here the multi lines was developed by the Borlaug against rust in the wheat. So here MLKS 15 was developed at the IARI. And next is new plant types for increasing the yield in case of wheat crop. And next is biotechnological approaches. These biotechnological approaches are also used in the wheat improvement or in vitro production of the haploids and also somaclonal variations. So these are the seven breeding methods in case of wheat crop, that is introduction, pure line selection, hybridization and selection, hybrid wheat, mutation breeding, and development of the multi lines and new plant types for more yield. And next is biotechnological approaches. So these are the eight breeding methods in case of wheat crop. And next, coming to the breeding centers, here we have three breeding centers. That is, first is a CIMMET. CIMMET is nothing but it is a the abbreviation for CIMMET is it is International Maize and Wheat Improvement Center, which is located in Mexico. And next is IIWBR, that is Indian Institute of Wheat and Barley Research, which was located in the Karnal. And next is AICWIP, that is All India Coordinated Wheat Improvement Project. It was located in the Karnal, but earlier it was in New Delhi. So this is about the wheat crop. Next, we will discuss about the oats crop, that is Avena sativa. This is having the chromosome number 2n equal to 6x equal to 42. This Avena sativa is also hexaploid, so it is having the chromosome number 42. And next, coming to the centers of origin. The centers of origin for oats, it is Asia Minor, and the progenitor for oats is it is Avena sterilis, and the it is a hexaploid wild species. And it is also having the related species which are the genus genus Avena was classified into 19 taxonomic species, which includes diploids, tetraploids, and hexaploids. So the diploid oat, it is having the chromosome number 2n equal to 2x equal to 14. And we are having the diploid weeds, that is the first is short oat, that is Avena brevis, and desert oat, that is Avena vesti. And next is sand oat or grey oat, it is Avena strigosa. And next is the small seeded or naked oat, that is the Avena nudibranchs. So these are the four diploid weeds which are present. The short weed is Avena brevis, desert oat is Avena vesti, and sand oat or grey oat is it is Avena strigosa, and next is small seeded or naked oat, it is Avena nudibranchs. And next is tetraploid oats. Here we are having the tetraploid oats, that is 2n equal to 4x equal to 28. Here we are having the two different types of tetraploid oats. First is the slender oat, it is Avena barbata, and next is Ethiopian oat, it is Avena abyssinia. And next is hexaploid wheat. So here we are having the chromosome number 2n equal to 6x equal to 42. So here we are having the 6 hexaploid oats. So first is a red oat. Here having here it is a Avena byzantina, and next is wild red oat, that is Avena sterilis, and next is common wild oat, that is Avena fatua, and next is common tree oat, that is Avena sativa diffusa, and next is common side oat, that is Avena sativa orientalis, and large naked oat, it is Avena nuda. So here the genus Avena was classified into 19 taxonomic species. So it having the diploid species, tetraploid species, and hexaploid species. Here the diploid species they are having, we are having 4 diploid species in case of woods, and here are 2 tetraploid species we are having, and next we are having the 6 hexaploid oats. Next, coming to the breeding objectives, the first is higher yield. In each and every breeding objective, the yield is the most important objective, and next is winter hardiness, and the third one is breeding for adaptability, salinity, water stress, and temperature, and the fourth one is breeding for resistant to the lodging as well as for the shattering, and next is breeding for disease resistance. In case of oats, the crown rust, stem rust, smut, and powdery mildew, and helminthosporium blight, that is bacterial blight, and barley yellow dwarf virus. So these are the most important diseases in case of wheat crop. And next is breeding for insect resistance. So here we are having the major pest in case of wheat are cereal leaf beetle and root knot nematode. And next is breeding for the quality. Generally, the green quality which is having the protein, that is the protein in oats is nothing but evanillin. And next is market quality, that is it should have clumpy and heavy grains. And for milling content, milling percentage, it is a growth percentage. And next is fodder quality. అమ్మ తర్వాత చూడండి తర్వాత చూడండి high high crude protein and nutritional quality, that is high protein with lysine content. So these are the some of the breeding objectives of oats crop, that is high yield, winter hardiness, breeding for adaptability, salinity, water stress, and temperature. And next is breeding for resistance to lodging and shattering. And next is breeding for disease resistance, and breeding for insect resistance, and breeding for the quality. So these are the 7 important breeding objectives in case of oats. And next is breeding methods. The first one is introduction. The Kent was introduced from the Australia. And next is selection, that is HFO 14. It is a pure line selection by CCS HAU, that is a Hissar. From Hissar they have introduced the pure line selection, that is developed the pure line selection variety, that is HFO 114. And next through hybridization method, that is OS6 7 and HJ8 and OS 346 were developed through hybridization and selection. And next is we are having breeding methods, that is a development of multi lines. And next is marker assisted selection. Here the marker assisted selection is used for development of disease resistance and milling yield and growth protein and oil content, and for the development of the short straw and panicle weight in case of oats crop. And next coming to barley crop, that is Hardium vulgare. It is having the chromosome number 2n equal to x equal to 14. So as we know, the barley is the most important cereal crop in the world, next to the maize crop, and maize, wheat and rice crop. In India, it is the most important cereal in the winter after wheat, both in area and production. And this barley is cultivated in the UP, Punjab, Rajasthan, Haryana, Madhya Pradesh, Bihar, and also in the plains of Himachal Pradesh, Uttaranchal, and Jammu and Kashmir. And the centers of origin for barley is, it is a Western Asia or Ethiopia, and the progenitor for barley, it is Hardium spontaneum. So for next we'll discuss about the breeding objectives, that is high yield. The main objective is high yield, and it is barley for used for the feed as well as for the food purpose. And the most important quality parameter in the wheat, it is malting quality. So we have to develop the varieties which are having good quality malt. And next is breeding for abiotic stress tolerance, and next is breeding for resistance to the both biotic stresses, that is pest and diseases. And next, coming to the breeding procedures, which includes both conventional as well as innovative approaches. So here we are having the seven breeding methods. That is, the first one is bulk method, and the second one is composite crosses, and the third one is male sterile facilitated recurrent selection, and the fourth one is pedigree method, and the fifth one is bycross method, and the sixth one is single seed descent method, and the seventh one is, it is haploid breeding method through bulbosum technique. So these are the seven innovative breeding approaches which are present in the barley crop. And next, coming to the summary, this lecture covers about the breeding, about breeding methods, about origin and the improvement of three major cereals, that includes the wheat, oats, and barley. Here, the wheat, it is a Triticum aestivum, which is a hexaploid staple food crop in the world, with objectives including the high yield, lodging resistance, disease resistance, and the quality improvement. So these were achieved by different breeding methods like pure line selection, hybridization, mutation breeding, and biotechnology. And next is Avena sativa. As we know, the scientific name is the oats of scientific. The word scientific name is Avena sativa. It is also one of the most important cereal for food and fodder, with breeding focused on the yield, winter hardiness, stress tolerance, and resistance to the rust and BYDV virus. And next is barley. This Hardium vulgare, and the scientific name of barley is Hardium vulgare. It is also ranks next to the major cereals, and it is also improved for developed for feed, malt quality, stress tolerance, and pest resistant using some of the conventional and innovative breeding methods. So this is about the wheat crop, oats crop, and barley crop. Here we have discussed in the wheat crop. The most important thing is, it is a scientific name, that is Triticum aestivum. It is a hexaploid species. It is having the chromosome number 42. And here we have discussed, it is one of the most important staple food crop in the world, followed by rice. And in every month of the year, we can see the harvesting of the wheat crop in the world. And here the origin is Southwest Asia for the wheat crop. And in wheat, we are having three different ploid species, that is a diploid, tetraploid, and hexaploid. For tetraploid, it is 14 chromosome number. For tetraploid, it is 28 chromosome number. And for hexaploid, it is 42 chromosome number. And next is, and here we discuss about the cultivated species, that is about Triticum aestivum and Triticum durum. This Triticum aestivum used for making the breads, cakes, noodles, and cakes, and cookies, and chapatis. And whereas a durum wheat is used for making the flatbreads. Here the Triticum aestivum is the hexaploid, and Triticum durum is the tetraploids. And we are having the 16 wild species, out of which the two are diploids, and seven are the tetraploids, and three are the hexaploids we are having in the wheat species. And based on the quality, they are divided into hard wheat and soft wheat. So here hard wheat are tetraploids and soft wheat are hexaploids. And here we can see the protein content around 13 to 16% in case of hard wheat, whereas in soft wheat we are having the protein content of 6 to 11%. And this is the durum wheat, and the soft wheat is the bread wheat. And these are glutinous and these are non-glutinous, and these are used for making the marconi, and the soft wheats are used for making the cakes, pastries, and cookies. So here we have discussed about the origin of three diploid ploids, that is a diploid wheat, tetraploid wheat, and hexaploid wheat. Here you can see here the we obtained the triticum monocom from the triticum boiticum. This boiticum is the ancestor for all the cultivated wheats. And next is hexatetraploid wheat. This triticum monococum is crossed with unknown species. We got the head diploid. Again on the chromosome doubling, we got the triticum turgidum, that is amphidiploid. And next is origin of hexaploid. This triticum, this tetraploid, that is triticum turgidum, that is crossed with tauchi, that is diploid species. We got the triploid. Again, it was again the chromosome doubling was done with the colchicine treatment, so that we got the hexaploid species, that is triticum aestivum. It is having the chromosome number 2n equal to 6, 6 equal to 42. Here it is a fertile and having the genome AA, BB, DD. And these are the five breeding objectives which are most important in the breeding in the wheat. And these are the breeding methods which we have discussed. And these are the three breeding centers which are present in the wheat crop. And this is a oats. Here oats we have discussed about the centers of origin, and we are having around 19 species, which includes both the diploid, tetraploids, and hexaploids. In diploids we are having four diploid oats we are having, and here we are having two tetraploid oats and six hexaploid oats. And these are the most important breeding objectives in case of oats. And these are the breeding methods which are in a breeding methods in case of oats. And next is barley. This is also most important cereal crop next to the maize, wheat, and rice. This is a most important cereal in the most important cereal in winter, after wheat, both in area and production. And these are the breeding objectives. In barley, the malting quality is very important. And next is these are the breeding procedures. That is the conventional and innovative approaches. So here we are having seven breeding methods in case of barley crop, and this is a summary.